Good morning, everyone. So it's my great pleasure to be here today to tell you how we explore the potential of nanopore sequencing to transform cancer care, both at the germline and somatic levels. So within the past years, there has been a huge improvement in sequencing technologies and tumor understanding, which has led to a complete transformation of the way cancer patients are managed, while also having an impact on their relatives when a predisposition is identified. So in this context, we were interested to see how nanopore sequencing could add value to this new definition of management. So at the lab, we studied it both at the germline level, with the structural variance characterization, and also at the somatic level, keeping in mind a continuous, better, and more precise characterization of brain tumors and sarcomas. Indeed, the Curie Institute is one of the reference centers for pediatric brain tumors and sarcomas, so I will highlight it a bit later in my talk, as we will start with the germline structural variance characterization. So, as you may know, in oncogenetics, the identification of a pathogenic germline alteration has always been a fundamental, as it will guide not only the immediate patient's management, but also their relatives. So within the last year, with the appearance of next sequencing technologies and multi-gene panels testing, there has been an acceleration in the detection of single nucleotide variants. However, regarding the structural variants, this is still a challenge. So in this context, we wanted to see how nanopore sequencing could overcome it. So I will not remind you how adaptive sampling works, but this was just to tell you that this is the approach that we chose, as we wanted to selectively enrich on some genomic regions of interest. And we were working on MK1B device, which is more than enough to obtain the data that we need. So this is the workflow that we developed that is mainly based on the recommendations of Oxford Nanopore technologies. So we prepared the libraries using the long sequencing kit 114. And as you may already know, the preparation of the library is really fast and easy, and it allows us to perform it within half a day. So after this, we're going to inject the library into two different parts. The first part will be injected on the MK1B for almost 24 hours. After this time, we're going to stop the run to wash the flow cell and reload the second half of the library for another 24 hours. So after this, the bioinformatic analysis will be automatically launched. So choosing adaptive sampling, we developed two different approaches. The first one on the left is when we want to study a panel of genes of interest. So in this case, we're going to load into the MinNow software all the list of the genomic regions of interest, and we take into account 5 kb of linking region upstream and downstream each gene in order to increase the coverage. So we try to obtain the files representing between 0.1% and 10% of the genome, and the closer to 1%, the better. The second approach, as you can see on the right, is following initial analysis, or depending on the clinical context, when we want to look at one particular gene, for example, the BRCA1 gene on the long arm of chromosome 17. In this case, we're just going to enrich on the long arm of this chromosome in order to have a comprehensive overview of this genomic region. So as I was telling you before, the bioinformatic analysis is then automatically launched. The pipeline is called NanoClead, and it's freely available on GitHub. So if you are interested, I invite you to go and check at the link on the slides. It was developed by our bioinformatician team, and I will not enter much into details, but this was just to tell you that for the SV calling, we use a combination of Sniffles2, Zvim, QTSV, and Nanovar, and we're going to review the reads manually using Integrative Genomic Viewer. So with this approach, of course, we're going to be able to characterize structural variants, but we will also have access to the methylation information, as there is no treatment of DNA or amplification steps. And this is particularly interesting when we want to focus our analysis on some promoter of tumor suppressor genes and to look at the methylation there. And finally, as we will perform long-read sequencing, this will also allow us to perform variance phasing. So for today, we're going to focus on the structural variance characterization. And as you can see here on the slides, since we developed this approach, we have been able to characterize many rearrangements. And for example, we could see the insertion of mobile elements in the exonic regions of tumor suppressor genes, which were completely missed by short-read techniques. And also on the top right, you can see that we were able to precisely characterize a pentaplication. And so this highlights the interest of long-read sequencing to differentiate between a duplication and a more complex one like a pentaplication. So to be a bit more concrete, I will present you one example that we published 1 year ago, which was the case of a woman before the age of 61, presenting with a triple negative ductal carcinoma of the breast. Her medical records reported tobacco and alcohol consumption, but as you can see on the family tree, her family was not really suggestive of a hereditary breast and ovarian cancer predisposition. But following the French recommendations, as she presented with a breast cancer before the age of 61, she was prescribed a HBOC multigene panel. So this panel came back with the result of a heterozygous germline duplication of BRCA1 exons 18 to 20, which was subsequently confirmed by MLPA. But once we had this information, we could not assess the pathogenicity of this variant because we didn't know where in the genome it was occurring and basically if it was a tandem duplication or not. So normally in this case, this would require RNA analysis, which means another sample for RNA extraction, and another technique that is not routinely available in our lab. So here, given the context, and because the clinicians needed a fast answer to know if the patient needed a surgery or not, we decided to perform adaptive sampling. So we prepared the library, as I told you before. This was just to precise that we were working on the latest chemistry with the R9 flow cells. And so after we obtained the results within 1 week, and as you can see here with the reads on IGV, it was really fast and easy to confirm that this duplication was indeed a tandem one, because you can see that the colored reads align twice on the regions involving exons 18 to 20 of BRCA1. And so with this sequencing, we were also able to detect with a very good precision the breakpoints. And this allowed us to perform Sanger sequencing to confirm this event and to classify this variant as pathogenic. So within 10 days in total, we were able to go back to the clinicians and confirm them that she was carrying a pathogenic variant in BRCA1. And this was really important because another patient was eligible for surgery. So from our experience in the germline structural variance characterization, nanopore sequencing is really a fast and efficient tool to characterize complex variants that can be missed with the conventional short-read techniques. And this is performed within a really reduced time frame, which is compatible with clinical decisions. So now we will go to look at the somatic level, and we will start with the simultaneous methylation and fusion detection in pediatric brain cancers and sarcomas. So in neuro-oncology, there are many entities that are challenging but crucial to differentiate because they will induce completely different diagnoses, prognoses, and treatments. And within the past years, the study of DNA methylation has proven its undeniable impact in the help of classification of those tumors, and it has now become a criteria of the WHO classification of brain tumors. And so currently, this study is performed using Illumina EPIC array. And so with the methylation criteria, there is also the appearance of other molecular criteria involving genes of interest that will be important to study at diagnosis in order to obtain the most precise classification of the brain tumor. And so in the case of brain tumors, for the methylation analysis, we're going to rely on the classifiers of the team of Kapper in Heidelberg, which comprises 82 different tumoral entities, which they studied using the Illumina EPIC array. And thanks to this classifier, when we're going to study our tumor of interest, using random forest algorithm, we're going to be able to determine to which tumor group it belongs. So for this study, we also chose the approach of adaptive sampling, and this time we created the panel of genes with all the genes of interest in those tumor entities. So as you can see here, we also took into account 5 kb of flanking regions for a total size of 1.6% of the genome. And so this is really a three-in-one tool for the classification of brain tumors and sarcomas. Because of course, with adaptive sampling, we're going to be able to study the fusion with the list of our genes of interest. But we will also use the information of the rejected strands, which will be the small strands not on the genomic region of interest. And we're going to use this information first to build the whole genome copy number profile, but also to obtain the methylation profile, because we will look at the information of the methylation of the CPGs genome-wide. So since we developed this approach, we have now characterized more than 1/10 of frozen brain tumors with a complete overview of the tumor dynamics. And as you can see here, we were able to characterize various types of tumors. And so once again, in order to illustrate a bit this, I will show you some examples that we met in our practice. So the first case was a young girl presenting at 1 year old a bulky right hemispheric tumor. The histologic aspects were compatible with an ependymoma. So we received the frozen brain tumor and we performed adaptive sampling. So first, as I told you, we were able to obtain with a very good precision the copy number profile in order to study the dynamic of the tumor. And as you can see here, we detected a chromosome 22 monosomy. But we were also interested, of course, in the results of the methylation classification. And here you can see the t-SNE using the classifier of Kappa, where one color is one tumor group. And so in this case, the tumor was clusterizing with the ependymoma presenting with a YAP fusion. So with this information, we went to look at the YAP gene from the enriched reads of adaptive sampling. And as you can see here on this screenshot, we were able to detect with a very good coverage a fusion involving the YAP1 gene and the MAMLD1 gene. And so this information, combined with the methylation, allowed us to conclude that this was indeed a YAP1 fused ependymoma. The second example was a 6-year-old boy presenting with posterior fossa lesion, but at the anatomopathology, they were not able to distinguish between a medulloblastoma or an Ewing sarcoma. So once again, we received the frozen tumor, and we performed adaptive sampling, obtaining, as I told you before, the copy number profile, still with a very good resolution, and we went to look at the methylation. And in this case, we used the classifier of the team of Kulch, which is the same principle that the one of Kappa, but where they gathered the groups of sarcomas. And as you can see here on the t-SNE, we were able to confirm that this was indeed an Ewing sarcoma from the methylation. And this kind of tumor is characterized by a typical fusion that involves the EWS01 gene. So we went, of course, to look at this gene with interest, and we could detect, indeed, the fusion involving EWS01 and the ERK gene. So once again, the methylation guided us, and the targeted reads confirmed that this was indeed an Ewing sarcoma. And so we were really happy to see these results, because the potential of nanopore sequencing for brain tumors has already been described, but this was not the case yet for sarcomas. So for this part, from our experience, we really think that nanopore sequencing is a really robust three-in-one tool for the classification of brain tumors and sarcomas, because it allows us to obtain simultaneously the copy number profile, the methylation profile, and to detect the fusions of interest in those tumor entities. And all of this is performed within a really reduced time frame, which was around 1 week from our experience. And so to conclude, I will have a quick word about medulloblastomas, grouping, and subgrouping. So medulloblastomas are the most frequent embryonal brain tumor in the pediatric population, with a mean age of diagnosis of 6 years. They are divided into 4 groups, which are the medulloblastomas WNT, SHH, group 3, and group 4. And once again, depending on the group, it will involve completely different prognoses and treatments. So this highlights the need for an efficient grouping method. So as I told you at the beginning, currently the assignment of methylation is performed with the Illumina EPIC array. So these arrays are going to study the methylation at more than 800,000 CPGs. But however, the technical preparation can be a bit complex, and it's time-consuming and expensive, and not always suitable for an emergency situation. So in our study, we were interested to compare the results of methylation obtained with whole-genome nanopore sequencing, either by multiplexing on a MinION flow cell or one patient on a fungal flow cell, and we compared those results to the methylation results with the Illumina EPIC array on 40 frozen medulloblastomas. So this time, we didn't perform adaptive sampling, but we just performed whole genome nanopore sequencing, which allowed us to obtain first the copy number profile. And as you can see here on this example, it allows us to detect the amplifications of interest, notably in the MYC gene for the medulloblastomas. And also, of course, we wanted to obtain the methylation profile. And so the results that we obtained speak for themselves, because you can see on the left the results of methylation given by the EPIC array, and on the right, the methylation given by nanopore sequencing. And so we are really happy to see that both results were really concordant between both techniques, and that nanopore grouping reliability is similar to the one of the gold standard. After this, we wanted to be a bit forward and to go a bit forward and to validate those results on another cohort of 116 medulloblastomas that we studied again with whole genome nanopore sequencing. And this time, we compared the results to an integrated diagnosis comprising immunohistochemistry, short-read panels, nanostring, and anatomopathology. And so once again, we were really happy to see the results that we obtained. We had only 6 samples that were discordant between both techniques, but globally, the grouping given by Nanopore was concordant with all the clinical and molecular information that we previously gathered. And so to finish, we wanted to go even more further and to go back at our first cohort, because now for the medulloblastomas, it has been proven that there are subgroups in each group, which involves, again, different prognosis and treatments. So we wanted to see if the methylation information given by Nanopore could allow us to obtain the subgrouping of medulloblastomas. And so our preliminary results are really encouraging, because it seems that indeed the Nanopore methylation results allow us to obtain the subgrouping of medulloblastomas. So for this part, we were really happy to conclude that the grouping reliability obtained with nanopore sequencing is comparable to the current gold standard, and that we could also have access to subgrouping of medulloblastomas. So this is really an accessible technique which is easy to implement and is particularly suitable for emergency situations, because we could imagine loading just one tumor on a fungal flow cell and obtaining the results of classification within 24 hours. So to conclude, from our experience, we really believe that nanopore sequencing is a versatile tool with a wide range of applications from the germline side to the somatic level. So it offers a characterization that is increasingly fine-tuned and which is rapidly adaptable to the situation. Because with adaptive sampling, if we just want to study another region of interest, we're just going to change the file that we load into the MinNow software, and it will not involve designing a new panel of genes or creating primers for sequencing, so it's really adaptable. And so at both the tumor and the germline level, it really has the potential to revolutionize patients' management through a personalized approach. Thank you very much for your attention, and I would like to thank all the team of the Dr. Julien Maslia-Planchon in the Curie Institute, who are working on me on this project from the wet lab to the bioinformatics pipeline. Thank you very much.